All right. Hello, everyone, and thank you for joining us this evening. While we all get connected, I'll just introduce myself briefly. My name is Rachel Skipper. I work in admissions at Syracuse for the College of Arts and Sciences and the Maxwell School. But I'm not the star of today's show. That is Professor Hewett, who is also with us here on screen. Our class today is going to focus on neuroscience and specifically the different types of cells that support brain function. And leading our class is Professor Sandra Hewett. Professor Hewett is a Syracuse professor, very popular among our undergraduate students, as some of you already know. So I'm excited that you get to meet Professor Hewett today and see a great example of what our undergraduate students experience in her classes. She's also our executive director of neuroscience studies, meaning that she has an important role in developing and maintaining the strength of our neuroscience program and the quality of the program for people like you who we hope will enroll in the program in the future. She also has an active neuroscience research lab on campus. So if you have questions about undergraduate research or about what mentorship looks like between faculty and students, how you develop research projects and how we answer research questions as neuroscientists on campus, then Sandra Hewett is a perfect person to ask. You can find many examples of her mentorship on the Syracuse websites. And I'm happy to connect you with students who have worked with her in the past. They all have great things to say. And if you want to hear about research from a student's perspective, we can do that after the session. So with that being said, thank you for being here, for Professor Hewett, and I will let you take it away.
Thank you so much. I just can't believe how many of you guys are so interested. I'm so excited that you're interested in Brain Blast Beyond Nerves, but also Syracuse University. As you can see, it's auto. We love auto and we bleed orange. If you don't know that, and it'll be fun after this, I'm happy to take as many questions as you want on Syracuse itself, on the neuroscience program, how students interact and why we're such a great community here. But yeah, today when Rachel asked me to meet with all of you, I thought, I think everyone kind of understands the term nerves, but in my class, so some of this is literally been taken from my class, which is actually the advanced neuroscience class. So if you come here and you become a neuroscience major, I teach the advanced neuroscience, which is you will take an intro neuro. And what I like to tell students are, everything that you learned at intro neuro, I'm going to blow your mind. I'm going to make it it's going to be in more detail. You're going to learn a lot more things. And one of the things that I absolutely love to do in my class is to say, absolutely, I cannot be talking to you. I can't be moving my hands the way I am. Sorry, I do talk with my hands. Without nerves, right? Without neurons, which is a single nerve, and many neurons make up a nerve. That's what I will say. We couldn't do any of that without it. But our brains, and I'm going to focus on the central nervous system today, although we're going to touch upon the peripheral nervous system, they could not function without the other cell types that are just as important. And these are really ripe areas. So some of you who are interested in neurology, some of you who are interested in neuroscience research, studying nerves are great, or neurons are great, and we do that in my lab, but we study what I call the neuron astrocyte, and you're going to learn about that if you read about brain blast interactions. So nerves could not function without these cells. So I wanted to give you a little flavor of what else is in the brain and why these are also important, and show you some really cool pictures that I hope will, because every time I show this picture of the vasculature, everyone goes, oh! And so will have, I guarantee you when you leave today, and it will only be about 20 minutes that I'm going to talk, and then you will have an opportunity, you will think about your brain very differently. I guarantee it. Okay, so here we are. So do you guys want to be a little interactive? Does anyone want to do chat or unmute themselves and talk to me? Because I know there's 100 of you, so it's a lot of people. But someone says yes. Okay, so can anyone tell me one of the main functions? What do you think are the 3 main functions of the nervous system? Anyone?
Letting the body respond to stimuli.
Oh, that's one. But before, we have to do what? So we are going to respond to that stimuli. But before, somebody's in the chat. I'm going to look. Autonomic processes. Yep, those are automatic, but it will do that. I can do that. So we have to respond. We have to what? We have to sense it first. So I'm going to go, We have to gather the sensory information. So like if you just think of the nervous system, synthesize that information and then respond. Thank you so much, whoever said it. So we gather information, sensory information from our environment, right? It could be our outside environment. So I'm very cold right now because my office is cold. But it could be our inside environment. Some of you might be like, oh, my bladder is full. It is very much time for me to go take a break, right? So we have both external and internal sensory systems. This information gets synthesized, if we're talking about our muscles, through and back up to the brain, where we're going to decide what we need to do and then respond. So I've also made, I don't know why here we go. I've made this small little one to kind of say this, right? So I always joke, like, here I see a donut because I like donuts. So I see a donut. I understand it's a donut. I integrate that and I say, I'm going to go walk into my kitchen, pick up that donut. This is a foot. So I'm going to walk into the kitchen, pick up the donut. And that's really what we do. So in this case, I chose the eye, but we have sensory input. Our brains integrate this. It goes through our spinal cords, back out, and forms motor input. So that's the central nervous system is our brain. Oh, I was going to not do it, so I'm not going to do it. So we have communication between our central nervous system and our peripheral nervous system. So in my next slide, I was going to say, so I just said central and peripheral. Does anyone know what makes up the central nervous system? Shout out, there's two things.
The central nervous system is the brain and spinal cord.
Yep. And Sophia wrote that too. So exactly. So is the brain and spinal cord. And so our brain processes that sensory information and coordinates. And someone talked about that autonomic function, our conscious, that's me picking up my water bottle. I consciously want to drink, and our unconscious bodily functions too, right? Breathing. Right? I don't have to think about breathing. If we had to think about breathing, we'd be in trouble. And the spinal cord directs that signals, right, from the brain out into the periphery. Okay. Does anyone think, this one's a little bit hard, so I'll give you the peripheral nervous system, but the peripheral nervous system is really made up of our cranial nerves and our spinal nerves. And so those integrate, that's from like the spinal cord, integrate where those are. So brain, face, neck, torso, or out further along. Okay. And we won't get into it, but there are different transmitters, right? These are the molecules that transmit, that occur lots more in the central nervous system, some in the peripheral nervous system. But that's really what we have. I'm going to try to monitor the chat at the same time. Yep. And I had another person who wrote that too. All right. So this is going to be, I'm only going to talk about neurons for just a slight little bit so that we get on the same page. But this is what we would call our model neuron. Okay. So if we have a cell, it's polarized, right? Because like different from each sides. And some of you might have had neuroscience already or know something about neurons, but it is a cell, just like any cell. So it has a soma, right? It has a cell body, but it has these very specific processes that we call dendrites. And this is where signals from another neuron, and I'll show you this when I do this, would send chemical signals. They will bind here. And this is how we change our chemical signal to an electrical signal. Okay. That electrical signal travels down this axon to what we call the synaptic boutons or the terminal buttons. And this is actually where we store our neurotransmitter. And so if this signal makes it all the way down, our electrical signal, it's called an action potential. If it makes it all the way down, it will trigger release of neurotransmitter. And if I happen to be, I have to move you guys out of the way. I think you might be able to see it. If I happen to be in the peripheral nervous system, I might be a muscle. So this would be what we would call acetylcholine, would bind to the muscle and cause it to contract. Okay. If I'm in the central nervous system, it's another neuron, like you can see here. So in this case, it's going to be a neuron releasing onto a neuron, releasing onto a neuron. And there can be multiple, and it's very, very complicated feedback loops. But this is just, if you're just thinking about electrochemical transmission, this is how it works. All right. But, oh, this is my last part. So that's what I say is our model neuron. Those are the things that you see. But these are actual neurons from the cerebral cortex. And our cerebral cortex is actually made up. Some of you may have had this, so you're like, okay, it's the outer layer of our brain, and it's made up of these different lobes, and these different lobes control different things. Like our frontal lobe is our executive function, arousal, our temporal lobe, hearing. In the back of our brain, the occipital lobes, is where vision is processed and things. But what I wanted to show you, because I always like to tell my students, is a neuron's not a neuron's not a neuron. So here, our cerebral cortex are actually made up in layers, and you can just see that they are very, very different. And the only thing that I'm going to bring up with this is that how they look is really how they function. So they have very different characteristics on when we call it firing, when that action potential fires, how fast it fires, how it responds to other cell types. And I just didn't want to leave you, because I'm one of those people, I hate it when I go, that's a neuron. It's not really a neuron, but these are real neurons. So we have basket cells, all different shapes and sizes, and they synapse with one another. They synapse in different layers, and this is how they communicate. Okay. The reason I brought that up is because I actually had it. I forgot I had it. So spike patterns, right? So this is how we measure in the nervous system. It's really an electrical current, how these cells operate, if you will. And we call it a neuron fires, but it's their spike activity. So you can see based on their shape that they have very different spike or action potential patterns. So this is a very fast spiking cell. This is sort of a regular spiking cell. This is what we call burst spiking cells. So it bursts, 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 gets quiet, bursts, 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 quiet. And you can really look at this in terms of, yes, its own geometry, but also what synapse is on it. And so the synapse is really where one neuron meets another, and there's actually a gap. Okay. And the reason I bring this all up is because, yes, this is where everyone gets so excited and they want to study neurons, but neurons couldn't do any of this, like I said, if they didn't have other cell types. And there are more than these, but I wanted to focus with you guys on this, is talking to you about the fact that and now we're going to just stick with the central nervous system. Okay. So we're really talking about the brain here. We have these cells called astrocytes, which I'll tell you about. We have cells called microglia. Collectively, these are part of the glial cells of the nervous system. And nothing could happen in our brains if we didn't have blood vessels. And our blood vessels are made up of endothelial cells. And in our brains, we have more capillaries than anything else. So you've all heard of like your aorta. Well, it has a big, smooth muscle. Well, our capillaries literally are single cell blood vessels, and they're made up of these guys called endothelial cells. Okay. And so why do we care about endothelial cells? Because this is what we have. And that's why I say our brains could not function very well without it. They make up the blood-brain barrier. Okay. And as it says here, this is a highly selective, and it's semi-permeable. It can't block everything, right? Because we need things to get into our brains or we'd be in trouble, but it separates the blood from our brain and spinal cord. And it does that. And oh, here's the one I'm going to blow you away. This is literally a human brain where all of the cellular tissue has been removed by a process that's called clearing. And this is what your vascular system in your brain looks like. So my brain right now, think about it, has all of this, all of these blood vessels running through it. So I know when you see those brains and you see them and you're like, oh, it looks kind of mushy and whatever, all of these cells are in. And that is because, right, shout out to somebody, what do we need? What are the two biggest things you can think of that we need to get into our brains in order for all of our cells to function properly?
Oxygen.
Blood.
Oxygen? No, we never want blood in our brains. So these guys circulate blood to our brains, but separate it. So that's this whole idea. It separates our blood from our brain and spinal cord. Blood in our brain is very, very bad. And that would be what we call a hemorrhagic stroke. And those are 50% fatal. So I always say our bodies are kind of flawed in some ways, because why would you do that? But it's sort of the highway system to do it. Someone told me oxygen. What else?
Glucose. ATP, like glucose or something like that.
Exactly, glucose. So those, but I'm going to show you so much more. But of course, we need oxygen and glucose, right? Because what? We have to make a lot of energy, right? So all of you who have had bio, you know that oxidative phosphorylation, or in our mitochondria, right? We need oxygen and glucose in order to make ATP. So our brain consumes, like my brain might be 3 grams, right, of my entire body, and it consumes 20% of all the energy produced, right? Because as I wrote in my Beyond Brains, this is our minds, right? This is really what it is. It's our minds. I showed you this, and this is an incredible amount of blood vessels. Our vascular surface, our blood vessel surface, is about 20 meters squared. So let me put this in a way that you could see. Think of a 1 by 1 meter tile, which in our world is 3 feet by 3 feet. Okay, so think of a 3 foot by 3 foot tile, and you have 5 of them going up and 4 of them going across. Right? It's a pretty darn big area. That's the area, if you were looking at all this vessels, that takes up our vasculature. And then I told you about capillary, right? Is that single endothelial cell. That is it. These cells are in very tight apposition to each other. It's very different from other parts of the body. And I will tell you one of the reasons that is in a minute. But in order to bring that oxygen and glucose and the amino acids that we need, right, and all of the nutrients that we need into the brain, that the brain will allow us to have, there's a capillary within 25 microns of every brain cell. The width of a dime is it's a little over 1,000 microns. So think about that. That's how close they are, right? So a dime, if you looked at it, you'd be like, that's pretty close. Think 1/4 of that size, and there's a capillary and a brain cell, capillary and a brain cell. So our brains cannot function without this system. And like I said, it's a barrier, right? A semi-permeable barrier. And I always thought this is really kind of a fun thing that I showed, is a little bit of history. So this scientist named Paul Ehrlich in 1885, he was one of the first to determine that there must be what was coined a few years later, the blood-brain barrier, because he put a dye into, and this could be a mouse, but I'm sure he used a rat, but into a rat. And what he found was it was excluded from the brain, right? And so this is just a picture. Now, Paul Ehrlich got lucky, because if you do a small enough dye, and if that dye is what we call lipophilic, if it's oil loving, it can get in the brain. But he got lucky in 1885, and he was like, hey, there must be this mysterious barrier. And then I don't know why he didn't do the opposite experiment, but Edward Goldman did in 1913. He decided to put the dye in the brain, and lo and behold, what he found was it didn't go into the body. So this protects the things from the brain from going out unless we want it to, because we do have sort of a lymphatic system. It's called a glymphatic system in the brain that is necessary to remove waste. We absolutely do. But it also prevents things from getting in. And so the 3 main functions of that blood-brain barrier I list here, right? I kind of told you, with this tight apposition of these endothelial cells, things can't get in between it. It acts as a physical barrier. It is also a transport barrier in some ways, in a good and bad ways. So there are systems in our endothelial cells that allow their transport proteins, for real, that allow what we need to get in, like amino acids, and they transport them in. But our brains also have evolved, or our endothelial cells have done this, where drugs, and this is a problem. So these are the things we talk about in our class. So chemotherapeutic drugs can get into the brain, and we have these efflux transporters that say, no, we don't want them in the brain, and they pop them back out. So these are, some of the things, like, it's very good because it protects our brain, but it's so good that sometimes it prevents the ability of therapeutics to get in. And then it's also a metabolic barrier. And what that just means is within the endothelial cells, sometimes substances get into the cell and they metabolize. They have enzymes that will say, oh no, we don't want that to get in the brain, and we're going to metabolize it. So those little cells in close opposition are so important for our brains to function, and they act as these barriers. And I wrote here some of the things that we already just talked about. So that, but it allows glucose to go through a transporter, and I'll show you. Oxygen freely diffuses across the endothelial cell. Vitamins, amino acids require transport. Okay, so this is just a little graph that, like you might see, we call this a kind of, if I were doing it, it was a single drug, it would be a dose response curve. But what this group did here is they looked at what could get into the brains and what the properties are in some ways. So if we look at the bottom down here, we see what says low lipid solubility. So think lipids, think oily, right? So if I want to dissolve in water, I'm not going to get across the blood-brain barrier very well. However, if I'm highly lipid soluble, if I like to be in the oil fraction, I will. And so some of these drugs you've seen, and so some don't make any sense. And I've just circled this here. So these are a bunch of amino acids, and they're really highly in the brain, but all of these have low lipid solubility. So why are they doing that? Because, like I mentioned, there are specific, our endothelial cells have specific transport proteins. There's so many transport proteins that recognize these amino acids and allow them, they transport them into the brain. Some things are very lipid soluble. This phenobarbital is I'm going to tell you, it's a GABA agonist, but it acts on our inhibitory receptors to dampen brain activity. Very lipid soluble. Codeine, methadone, these things will go into the brain very well. Now, then we have, caffeine gets in. I have different things. We have some things that you think should get in, like morphine, you've heard, and it will, but it has to be metabolized first. So the drug itself, not so great, but once it's metabolized, it can get into the brain. But roughly, when I teach this, I tell the students if it's what we like to call lipophilic, it likes lipids because our cells are made of lipids, it goes across. If it's highly charged or it's has low lipid solubility, it's not going to get across the blood-brain barrier unless the brain wants it to, like with amino acids, like with glucose. Okay. And this is great, but in some conditions, and I want to show you, and this is what can happen. Oop, I got a lot of people chatting, but I'll go back to the chats. Blood-brain barrier breakdown can occur in lots of different, so after stroke, this happens to be what a brain looks like when there was a focal epileptic seizure. So a seizure in this rat was confined to one side. And then what this group did is they added a dye that you can see on the left side here is excluded. And where this rat, following this rat's focal seizure, it resulted in the blood-brain barrier becoming leaky. And so we can measure that, and we can look at that in the lab and actually in humans as well. But this shows that. And then this brain was sectioned, so you could see that it was a cortical seizure. And in the area where that seizure occurred, the blood-brain barrier becomes leaky. This, in general, can be very bad. But let's go back to what I said before. Sometimes we want drugs to get in. And so we actually have a colleague here over in engineering who is working to try to understand how to temporarily open up the blood-brain barrier to allow for these chemotherapy drugs to get in, or drugs that can, chemotherapy just means it doesn't have to be cancer. It just means a chemical therapeutic drug for Alzheimer's disease or for Parkinson's disease and things like that. So under some conditions, blood-brain barrier breakdown, I just saw in the chat real quick that somebody's dad had an epidural hematoma. That can be confined to the dura space. That can cause a lot of problems too, but it's not necessarily inside the brain. But we sometimes want to break that barrier down so we can get things in. Okay. All right. So this leads me to my other cells. So here are our blood vessels, right? So we have these blood vessels. Things have to get into the brain. And how do we maintain that really tight junction that affects, that prevents it? Well, what I can tell you, the other cell type, remember that glia I said I wanted to tell you about? Astrocytes, one of my favorite cell types of all time. I love them. This picture literally shows you, here are some blood vessels, some capillaries. Astrocytes are these are what we call their end feet in yellow here. Anything that you see in yellow is an astrocyte, and it encircles. 90 to 95% of our blood vessels are encircled by these astrocytes, okay? And these astrocytes in development, we know, are necessary for actually contributing to the induction. So getting these endothelial cells to be very tightly joined, and the maintenance. So if you disrupt astrocyte function or astrocytes die as well, it will alter the blood-brain barrier. Astrocytes also release what we would call vasoactive substances. So when our brains get activated, because I'm going to show you a picture, I think on the next slide, where the astrocytes are also relation to neurons, they will, when neurons are active, we need more energy, and that will be, some compounds will be released or chemicals will be released by the neurons, but the astrocytes will also respond to this response and release substances onto the blood vessels to make them dilate. And when that happens, more blood gets into our brains. So when we're thinking really hard, or like maybe that's happening with me now, certain areas of my brain are very much more active, then my blood vessel dilates so I can get more oxygen, more glucose to maintain the energy needed to keep my neurons firing. So neurons, obviously, like I said, it's beyond nerves, are really important for sure. But again, I hope what you can appreciate is they really can't function very well if the blood-brain barrier is leaky. It might let substances in that are toxic. It can let in bacteria, viruses, and so there's this coordinated activity between our neurons, and in this case, our glial cells and our endothelial cells, okay? And also, what I wrote on the slide here is that astrocytes, because of where they are, which I mentioned that glucose gets in, but it needs a transporter. Well, endothelial cells have a transporter that transport it into the brain parenchyma, we call it. Astrocytes have transporters that pick it up. And I'm going to show you on this slide, they actually provide energy substrates to the neurons. I know this is a lot, but I'm just going to show you, like, this is the souped up version. You don't have to worry about it. I'm going to give you the 30,000 view of it. But so here we have our capillary with our glucose, right? In it, we have a transporter called GLUT1, glucose transporter 1. It will transport out that glucose. Our astrocytes also have glucose transporter 1. It'll take that glucose into the astrocyte. Now, astrocytes need glucose, right, to make energy because they have to function too. So they use some of it to make energy and to maintain our gradients so that our membrane potentials are good. But they also convert it. They can convert it through, does anyone know what glucose to pyruvate is? Does anyone remember what this process is?
Glycolysis?
Yes, glycolysis. Yay! So glucose goes to pyruvate, and normally pyruvate would then go into the mitochondria, into the Krebs cycle, we call it, or the TCA cycle, to create energy. But astrocytes can convert this to what I call an alternative substrate, but it's really a great substrate, lactate. You guys have probably heard about lactate in your muscles, that lactate causes your muscles to get sore because you make too much lactate. Well, lactate is actually an energy source. So in this case, the astrocytes shuttle that lactate out through another transporter. Neurons pick up that lactate. They take that lactate and shuttle it back to pyruvate because the enzyme that does this is bidirectional. And then that pyruvate goes in like the normal into the mitochondria to make ATP. So astrocytes are so important for energy metabolism. Is really like big thing. But I wanted to show you how from going from the capillary to that astrocyte, to the neuron. And the reason is because I'm going to show you astrocytes are also, not only are they just all sitting against your blood vessels, but they're interdigitated here, as you can see, with your neurons. So the neurons are all here in blue, and my favorite cell, astrocytes, are all these cells just sitting around here around the cell bodies, here around, in this case, I can't tell if it's an axon or a dendrite, but I'll call it neuropil. So these astrocytes and neurons are completely also. So I don't know if you knew that, right? So you think you're just a bunch of neurons, but you're not. And funny part is astrocytes actually outnumber neurons in the human brain. Okay. So estimates are 4, maybe up to 7 to 1. So you actually have more astrocytes in your brain than you do neurons. So they got to be pretty important, right? So in fact, they constitute, depending on which species, 20 to 50% of your total brain volume. Okay, so they're really important cells. I love them. And so this is the schematic version of this, which is, again, we have the blood vessel, the astrocyte takes up that glucose, gets it to the neuron. But this is my segue. I hope you appreciate my transition. That there are other cell types in the brain that are absolutely necessary, right, to maintain proper function as well. And those cells are also a glial cell type, but they're called a microglia. Okay. And these microglial cells, this is just a picture here of a microglial cell, are not quite as populous as astrocytes. In fact, it's thought it's a 1-to-1 maybe ratio of microglia to neurons in our brains. Okay. Does anyone know how many neurons we have in our brain? Want to guess?
Like a couple billion, right?
Higher or lower? Anyone want to go with that? A couple of billion? Who says higher?
Higher.
Higher. 100 billion, close. So that used to be what we told everyone. So someone in my chat who told me that. Not 200 billion, but actually 80 billion. So there's been a reset, and they actually were able to count them better. We used to all tell everybody there were 100 billion neurons, but there's about 80. We have about 80 billion. And take care of them because they are what we call post-mitotic. And that means that they don't divide. So unlike microglia and astrocytes, which can replenish themselves in your brain, and microglia do it all the time, neurons don't. Oops, I just whacked that. But anyway, they are about 15% of your total non-neuronal cell population. They're about 1-to-1, like I said, with neurons, and they're distributed through the CNS. And they are incredible because they're also very beautiful cells. They are absolutely necessary to maintain, and I put the word we would use, homeostasis or stability, when the brain is developing. So they literally will go in and prune our synapses so that the correct connections are made. They monitor, and I'm going to show you videos of this, and then we're going to end. They monitor how healthy our brains are. Are there any invaders of our brains? And they actually respond also when there's injury and disease. Okay. And the cool part is they can do all of that by changing their shape. And I'm going to show you this. And you can tell the state of the brain by what shape your microglia are in. That's cool. So here's a picture of, these are like normal microglia. And what I'm just showing you here is that they tend to occupy these distinct territories. They don't get in each other's way. You know, it's kind of really an amazing, I think biology is amazing anyway, but development is very cool too. So this is actually, you won't know, but this is an area of our hippocampus, which is the area where we learn in. And these cells in red are neurons. And here where you see anything that's green, these are our microglial cells. And you can see that they're interdigitated. Some are within the neurons themselves, the neuron area themselves, but they're all surrounding the cells. And the reason that they do that is because they are continually surveilling the brain. So they do this when they're, I know, like say, I'm going to call them ramified, so you can see how pretty these cells are. This is what we would call ramified or resting. I don't think microglia ever rest. But they do this through this process called pinocytosis. So they literally sip the extracellular fluid and make sure that the ion concentration's correct. There's no invaders or whatever. And I'm going to show you a video of this, I hope. Yes, I am. So here, these are microglia in a mouse brain. The reason you can see them is because they were genetically engineered to express these would be green, a green fluorescent protein using a marker that is specific for these cells. So the only cells that get marked in this brain are the microglia. Now, if you take that mouse and look at its brain, you wouldn't see green. You have to see it under a microscope, a fluorescent microscope. But what they did, because I think this is really cool, is they just made, this is a living mouse brain, by the way, they made a little cranial window and then put this mouse under the microscope and just watched what's happening with these microglia. And this is what's happening. This goes a little fast, and then I'm going to zoom in. But I hope what you can see. So all of these, I'm going to stop it, or if it goes again, I'm going to stop it here. All of these are the processes that I showed you, right? So when you see all this movement, this is highly sped up because I'm going to show you on the next slide. It's actually a pretty slow process. It takes minutes, if not tens of minutes. But what these cells do are they protrude and retract their processes to sip in this to make sure everything's okay in your brain. So that's what it sort of looks like in this condition. Here now we're focusing in just on one of these, okay? And so over time, you can see what those processes are actually doing. So, okay, think about this right now. That's happening in your brain right now. I think that's cool. So, but like I said, this is very sped up. So if we just look at the real data, what you can see is that it takes time. So it really, over a 45-minute time, though, you can see this P1 that we're measuring here, you know, it gets big, it gets small, it goes up, it goes down. It's a very slow process, but it is happening in your brain. And because of the way they're arranged, they're able to really cover your entire mass here. All right. So remember I told you that single cell? Well, that single cell is really pretty. It puts out those processes. Now there's an injury. Say you were hit very hard on your head. And now what's happening in your brain when all that pinocytosis is happening says, hey, there's something wrong. These cells will actually retract their processes and become active. Okay. And they do different things. I don't want to get into it, but this would tell me that there's something going on in this brain. And then even more would tell me they would become reactive microglia. And so what the same group did, but now you can see it in green because they didn't do black and white, was they filled a blood vessel with a red dye. So you could see it as a blood vessel. What you're going to see on this is they then used a laser to break that blood vessel. So blood is going to go out into the brain. Remember, I said that's very bad. And what I want you to watch are what happens to these cells. Okay. And then I'm going to play it a couple of times because what you're going to see is that it matters how far apart. But here, we're going to play this. So here's the blood vessel. Here's the laser. Oh my goodness, look at this. So what is literally happening are the microglia are coming out of the plane. They're coming from underneath, coming over, because what do they want to do? They want to contain this injury. They can take up the blood and they'll do it. These are really, in this state of reactivity, what we call macrophages, big eaters, if you haven't heard that. They can now not just sip, they can engulf things. Plus, they can release factors that will help prevent, to try. It doesn't work very well, we have to admit, because we know bleeding into your brain is bad, but you can clearly see that they can react to this injury. So I'm going to play it one more time, I think. Okay. This one went a little slow because I also want you to watch that these cells never move. but some of the other cells, this cell here in particular, moved. So it is reacting to that gradient, okay? So here, once again, we got our laser. Watch this cell. And other cells coming up in from here. So they are responding. So they're absolutely incredibly important. Oh, this scale, I would have to go back and look. It's not, it's small. So microglia are about like 10 microns. They're pretty small. That's a great question. But I would look. So anyway, the last slide that I just wanted to show you, and then I'll just take questions on anything that you want, is these microglial cells are so important because they surveil our healthy tissue. They, in small local damage, that active cells, they can release factors that will be protective. They might release survival factors, things like that. And then they can actually respond to that large insult that I showed you by becoming even more reactive. And so I'm going to take you all the way back to the neuron, because I love sort of this comparison. I showed you the picture, and you might be like, why did she show us that picture of all the different cortical neurons in the cerebral cortex and how they function based on their shape? Those are distinct neurons. We can't, a neuron has to be that neuron. Microglia are so versatile that they can assume the shape, and with that shape will dictate how they can respond to the environment. So they're very important. And so this comes to the end of my brain blast beyond brains, so that you have a sense that our neurons, which I'm not saying are not important, because they are, could not function without this compendium or orchestra of cells that surround them, that protect them, that communicate with them to make sure that the microenvironment is just right, so that we can be the best that we can be. And that is it for me. Oh, I had that last one. So let me stop sharing so I can see some of you. Here we are. So that's it. What can I do? Any questions, or in the chat, or even any questions? It does not have to be about my lecture. I'm really so Luke, I think Rachel Skipper, who's on here, will be able to get you the recording, because we are recording. So someone asked me, how does a brain bleed affect your neurons? The problem is it causes, so there are two things that can happen. The things that are in your blood are very reactive. And I don't know if you've had chemistry, have you talked about reactive oxygen species, or what's the one that I want? Sometimes my brain doesn't work. Free radicals. You guys have heard of free radicals, maybe? So brain, when it interacts with components that are, the blood will produce these free radicals, and that will kill the cells, but also it creates a mass effect. I always love to tell people, because we don't think about it. We're encased, right, in our cranium, right? If we have a big tumor, think about it. It's not like it can grow out of our skull, right? So what happens? It kills our neurons. The same thing with the blood. It's going to create a mass effect, and in that area, it's going to kill the cells, but it's also going to it's going to move, or what we call herniate, our brains and move it out of alignment. And so that's why bleeding into our brains is so bad. All right. I'm open. Anyone can just unmute themselves. Can we do anything to protect these hard workers in our brain? Yes. So you know what I say to my students? It's Thursday night. Don't drink too much when you're in college, because you're going to kill your neurons. It's just like anything else. Exercise, huge studies. Exercise and a good diet is the best that we can do. I tell my son all the time, please don't hurt your brain. He plays soccer for years. When he was in middle school, I got him not to head the ball. It didn't happen. He's varsity. And every time he heads the ball, all I think of is, because again, is this hitting here and his back of his brain hitting there and going to the front. Okay. And maybe it's not a concussion, but it can't be good. So protecting your brain, right? From concussions, from falling, all that stuff. Those cells are all going to take care of themselves. Let me see what say. If you have a nerve damaging injury and a couple of months, down the line when you are mostly healed, are those nerves completely healed? Maybe not. And the question is, if you, particularly in our brain, so in our peripheral nervous system, which we didn't talk about too much, we have some regeneration. It's not good in our spinal cord. It's not good in our brains. So I do say that, and I kind of joke, there's some saving grace. So there are only 2 places in our brains that make new neurons. So even when I said to you, like a neuron dies, it can't come back, right? It can't divide. In what's called the subventricular zone, and we make new neurons, and those can move. And in our, excuse me, that's my nose, right? In our hippocampus, which is good news, we make new neurons. And it's actually been shown that exercise, so people who have depression, if they exercise, and even antidepressants can increase that neurogenesis, is what we call, and those cells will then know how to reincorporate into the hippocampus. So that's good. But those are the only 2 places. So when you lose somebody, anybody know someone with Parkinson's disease? They have the tremors that, I don't know if you remember Muhammad Ali, but he has it, right? A lot. There's an area in our brains, it's the striatum. These are specific cells called dopaminergic neurons. And the sad part is our brains are very good. We call it, they're very plastic. So some neurons can take over function, and they are also redundant. So we have a lot of cells. By the time someone notices that they have what's called an essential tremor, they've lost 80% of their dopaminergic neurons. They will not come back. They will never come back. Okay. So that is the second leading cause of neurodegeneration behind Alzheimer's disease. Oh, do you want to ask me about memories, emotions, and tears? I am the wrong person to ask because I'm a biologist. And that is definitely a psychological, biological, but it happens all the time, interaction that absolutely that occurs. I do not know the answer to that. And I am happy to say, when you become a scientist or a doctor, it's okay to say you don't know. So working in my lab, or in any lab. So you guys are, neuroscience students, some of you. So do you guys know about the program, how it works? Can I tell you a little bit about it? Because it's unique compared to some other schools. So we have what it's called an integrated learning major. And it is a full-fledged major, but it is not a standalone major. So what we do here at Syracuse is you will pair that with, I have a slide I've just made because I'm doing our program review, but I'll just have to speak to it. But I think you'd be kind of impressed by it. You must pair it with another major. So I will tell you that the major that is most, so we currently have 209 neuroscience majors. 100 of them are also psychology majors. 64 of them are either biology, biotech, and biochemistry. They make up 64, so 164. But we have English majors. We have someone who's in Whitman because they're interested in, which is our business school, neuroeconomics. We have a journalism student because she's interested in science journalism. We have students in chemistry. So some of you, if you're interested in chemistry, we have a great medicinal chemistry program. So we have chemistry majors. We have people in anyone interested in exercise science. So exercise science majors are also neuroscience majors, public health, public policy, But those are not the hugest ones that we have. Like I said, the biggest ones are psychology. So you would come, and when you decide, so the fun part is you can say, I definitely know I'm going to do neuroscience, but maybe I'm not going to pick my second major until my sophomore year. So, you can do that. So why do we do that? You might say is because we want you. So let me just stick to psychology and biology with the recognition. I mean, if you go look at exercise science, my son's applying to exercise science right now. You have to take biology, chemistry. It's, it is a science. I just even lost my own train of thought. So the psych and bio, I know why. As a psychology major, we want you to understand those behaviors that you're studying. Like I said to you, the frontal cortex, the frontal lobe is important for executive functions. You're going to learn that in psychology. By the way, biologists, that's what you're going to learn in psychology, in neuroscience. Psychologists, we want you to understand what underlies that behavior. Okay. So it's really trying to go, like I like to say, from molecules to minds. That's what Syracuse University does. We go from molecules to minds. And so you biology, more, bent people will understand the functions all behind the brain, not just that this is neurons, they have action potentials, they make, this is what they do. And you psychologists will now understand the biology behind that. So that is why we do that here, because we feel that this is much better, more holistic view, right? That you're not just a psychologist, because it is neuroscience, right? It's brain and behavior, and you're not just a biologist. So this is what we do. And don't freak out when you get here. We generally like to have you take your, like your Psych 205 and your Bio 121, 123, things like that before you get into neuroscience. So usually students start in their second year, and we really require 5, I have to go do it too. It's 8 in total, 6 core courses that everybody takes. And then you have electives. And those electives, I'm not going to show you, I could pull it up, but can be in any of the ones that we have told you about, but can be any discipline. So we even have a linguistics major who's really interested in it. You can take linguistics, you can take communication sciences disorders classes, you can take philosophy of the mind. I tell people do it all the time, mind and machines. So you can decide and expand. But also, just so you know, biology majors, we have a lot of extra other courses in biology that are neuroscience. So we have the biology of neurodegenerative diseases. It's not required for the neuroscience major, but you can use it as an upper elective in your bio major. Okay. Psych, the same thing. You're going to take abnormal psychology. Students, our bio students could take abnormal psychology for their elective if they want. So you can actually, so if you're just saying, it's just these courses, you can actually create your degree with more neuroscience than we require. Right? Because your different majors will accept these different courses as upper division courses. You, of course, will have to fill your core courses for psych and bio or biochemistry, biotech. Those 3 actually have different core courses. But we integrate, and that's where they call it the integrated learning major, into those majors. And I spent a lot of time on that because I wanted you to understand that it is very different than if you go and just say, I'm a neuroscience major and here's what I have to take. You really have a little bit more freedom, right? Other than we say, we want everybody to have these courses, but after that, you are able to pick and choose. We have a pain and addiction class. We have cognitive neuroscience classes, that so that you can do that. And we have an advising system here that maybe is a bit overzealous, I'm going to say, but you will not be left behind if you will get a liberal arts advisor to help you with the liberal arts core, because we are a liberal arts school. And I believe in that even as a STEM faculty member more than anything. I went to a liberal arts school, small liberal arts school, Providence College, and taking courses in English and sociology. And, it's so important. It really does. Philosophy, it expands your minds. So you will have a liberal arts core advisor, you will have a major advisor. And in neuroscience, it might be the same. Like if it were me and you were a bio major, you might get me for both biology and neuroscience. But we do have 2 dedicated neuroscience advisors, 2 faculty members who will make sure that you can successfully and easily integrate the major into your degree. Okay, so research. We are all about research here at Syracuse University for our all of our majors, by the way. So we actually call it, we have a whole unit. It's called SOURCE. It's Syracuse, and you're gonna make me remember this. Syracuse, it's Undergraduate Research and Creative Endeavors. Syracuse, oh, what's the O? It's terrible.
Syracuse Office of Undergraduate Research and Creative Engagement.
Syracuse Office of Undergraduate Research and Creative Engagement. Why do we have creative engagement? You more science-based people think research, but creative engagement can be anything. So one of our, our public policy people do research, right? Their output is just different. Okay. We encourage everyone here to do research, everyone, English majors, if they want. You are not required to, but you will have the opportunity to work in a lab if you want. The reason I brought up SOURCE is because they are there to actually even provide you with funds, which actually helps me, right? So you work in my lab and we come up with a project. In fact, I have to review them by tomorrow at 11:59 p.m., but they are reviewed and faculty determine, oh, yeah, this is good. Yeah, we'll put $5,000 towards that project. So when you're working in my lab, that's really helpful, right? Because it takes resources to do that. Regardless of whether you get a SOURCE grant, though, faculty always take undergrads into the lab. We also have an honors program. So here I am. It's not just neuroscience. I'll tell you all about SU, but it feeds in. So right now I have 2 honors students in my lab who will be graduating in May. The honors program requires you to spend 4 semesters in creative engagement. Those students, of course, are spending those 4 semesters in my lab. They are right now, the first year, they learn techniques. They get acquainted with what are the questions we're asking in the lab. They come to lab meetings. Like I said, they learn some things. This year, they are bona fide spending their year on their project independently. So one of my students, who, by the way, was on one of these calls 4 years ago, which I remember, he stayed on and talked to me, ended up coming to SU. Now he's in my lab. Has an astrocyte project where he's interested in how arsenic, which is, it contaminates, believe it or not just in the third world, but our water table, how that affects astrocytes and astrocytic function. And so I won't get into it, but he is making, he's making these cells himself. He's maintaining these cells himself. He is running the assays. He is running the show now. And he will write a thesis at the end of this, and it is required of the honor students. In the biosphere, if you're not an honor student, and you don't have to be, we have a distinction program, which is very similar, only it's not honors, but it does require you to do that. What I love about it is it also is a course, a 1-credit course, where you will get up with your peers and tell them about it. So the first time you present, you're just going to tell them what your question is. The second time, you're going to give a little bit of background and some of the methods. And so this is stuff, by the way, that you will use. So Rachel made a joke about my students, but right now, two of my last undergraduates, besides many being in med school, one is a PhD student in neuroscience at Vanderbilt, and one's a PhD student in pharmacology at Penn, right? And being in the lab changed what they thought they would do, because they were like, this is really cool. And so, of course, it's dependent on the advisor. Of course, it's dependent on what you do, but you can and should do that. And if we don't have anything, and I'm not making this up, today I did it. A student responded to me, sent me an email today, told me she was interested in my lab, but told me she was interested in concussion and injury, which I do stroke, but not concussion. Upstate, which is literally, I'm pointing, but you can walk 5 minutes to Upstate Medical School, has a whole concussion center with 4 faculty members that do research. And I gave her the names of those. I know one of the guys. I told her to use my name, like Dr. Hewett told me to contact you. So you are not even just restricted from working here. You can go over to Upstate Medical. They have a whole neurobiology program. Some of their faculty members even come here. I like to have them come into my class and do what I call the cameos, and so that you get to meet them as well. So we have that interaction with Upstate as well, and we work with you to get you where you want to be. But you do have to be proactive, guys, too, right? So it's not like that. We want you to do some homework, but we want to facilitate that interaction for you. So I know that I talk way too much, so I hope that was that. I'll stop. Anyone else? I tried to cover the program. I tried to cover everything. Do you think differently about the brain? Yay. All right. Well, if you can stay on if you're shy, or if you want to leave now, it's okay. Because I know we had an hour. I don't know what we have. Oh, look at that. I'm even on time. So, but if anyone wants to stay on, I can just stay on for a few minutes if you want to chat with me. But if not, thank you guys for coming. Go Orange. Good luck with I know my son's doing the same thing. So good luck with your search. And I'm stressed. Hope it works out.
Thank you.
Thank you.
You're most welcome.
Thank you.
Thank you so much.
You can
Thank
and you
you.
can
Thank you
find
very
me
much.
online and send me an email if you want.